Hello, everyone, and welcome to today's live broadcast, Highly Specific Multiplexed RNA Imaging in Tissues with SplitFISH. I am Alexis Krause of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Andor. For more information about our sponsor, please visit their website at andor.com. So let's get started. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. I'd like to now introduce our presenters. Hao Chen, Senior Research Scientist, Genome Institute of Singapore, and Dr. Alan Mullan, Product Specialist for Microscopy Cameras at Andor Technology. For the complete biographies on our presenters, please visit the Biography tab at the top of your screen. Hao, you may now begin your presentation. Welcome, sir.
Thank you, Alexis, and thank you, Alan, for inviting me for this webinar. And it's my pleasure to tell you more about this project that the team has been working on, and it's this project about developing a method for highly specific multiplex RNA imaging in tissues. So let me begin. As an introduction, for the curious biologists, we are really interested in dissecting the tissue and cellular identities in our samples through the lens of gene expression, not just at the single cell level, but also we want to know, from an imaging perspective, where are they located in the cell? What is their spatial architecture and how are they organized within the tissue? And the goal can be described as spatially resolved transcriptomics. So transcriptomics is like the measurement of the gene expression or the RNA content of a cell. And the way to get more information from the gene expression is also to try to put them into their spatial context. For example, where each gene expressed in the tissue can tell us, the way the tissue is organized and what are the cell types in the tissue. And not just at the tissue level, but also at the subcellular level. If we can look at the localization of RNAs, perhaps we can infer their functions. How and what are they doing in the cells? There are currently many approaches that people use to achieve spatial transcriptomics. There are four that I highlighted here: multiplex FISH, in situ sequencing, sampling-based sequencing, or spatial RNA-seq. These different approaches have their strengths. And for this talk, I want to focus on this multiplex FISH approach. So FISH is like this technique where it's based on imaging. Basically, what it does is we use a lot of oligoprobes that are labeled, and then because they are complementary to the RNA sequence in the cell, the probes will target the cells and light up the individual RNA molecules. So this is an example of how it looks like. If you look at this image, under hybridization 1, with very high resolution imaging, you can see the individual RNA show up as diffraction limited spots. And the key idea of how we can do or resolve many transcripts in the cell is that we are doing this hybridization-based barcoding. What I mean is that we not only label the cell once, but we label the cell wash and label again. Of course, the sample has to be fixed in order for the RNA not to be moving around. But if we can do this many rounds, then we can image a lot of targets. And the way we do this is that by using this probe that has two ends to them, where we call encoding probe. We labeled the target RNA species, and then with two ends that are available for subsequent hybridization, then we can read out different groups of RNA. So the interesting thing about doing this sequential hybridization or serial hybridization is that the number of RNA species that we can probe actually scales very favorably with the number of hybridization, right? If we imagined, if we put the 1 as when we label this particular RNA in one group, and a 0 is when we don't label it, we can see from this binary scheme here that in a simple binary scheme, it scales exponentially with the number of hybridization. So with not many rounds of imaging, in theory, we can assign barcodes to all the RNA and detect almost the whole transcriptome. So in our earlier demonstration, we showed that with a different kind of barcoding scheme that is more error robust, we can detect up to 1,000 RNA species in the cell. So the way we confirmed that our measurement was accurate was that we counted all these dots from the cell, and then we tried to match them to known barcodes. And then we tried to compare the number of dots for each RNA species and compare that, for example, to RNA-seq here. And what we see is that the number of dots actually agrees very well with the RNA sequencing results. So with like, Pearson correlation of like 0.7, indicating that the dots that we are counting is highly reliable. In fact, with the method, it has been applied to many different areas, for example, to understand expression noise, or to understand gene network, or to do cell type mapping in tissues. It has many other possible applications in, constructing spatial cell atlas for multiplex diagnostics, pathogen detections, and etc. With this assay, we were trying to improve it and to apply it to different tissue samples. But what we found is that when we are trying to label a lot of transcript targets, sometimes the noise in these assays is very complex because, when we are trying to label so many targets, we use a lot of oligoprobes, sometimes like up to 100,000 different sequences, right? Then when we stain our samples, what do we see? We see like an image like this. It's a zoom in, maybe showing a cell region, and we see some diffraction limited spots, but it also has a lot of noise. And, we wonder where do these signals coming from? Are they real RNA, or are they from tissue autofluorescence, or due to like non-specific binding? And to optimize for binding for like so many sequences is truly very difficult, right? We can increase the probe length or try to narrow their melting temperature range, but when you use very long probes, they are actually not so specific, right? Because the thermodynamic penalty from a field mismatch, because the binding energy from a long probe will, outweigh the thermodynamic penalty from a field mismatch. So others have found a very clever solution that to reduce the noise in this assay, you can actually do gel embedding and tissue clearing. So what this does is you put your cell or tissue into a gel, and then you clear away all your other backgrounds. or rather you clear away all the proteins and lipids in the cell, and then that way the background is highly reduced. But this approach has some drawbacks, like you cannot remove non-specific bindings to other RNAs, and the clearing chemistry might not work for all tissue types, and sometimes the tissue clearing could take days to hours. So one way that we have sought to improve the specificity is to change the probe design itself. We have this idea that maybe we can improve the specificity by using coincidence detection. This idea where we borrow from essentially split GFP. So how this works is that in the cell, when you have two parts, the GFP doesn't fluoresce, even though it's at very high concentration, right? But only when it's on the target and the two GFP fragments come together, then the complementation then results in a full GFP that gives off fluorescence. So we want to see if this idea can be applied to oligonucleotide probes. When we look in the literature, actually the complementary or this coincidence detection idea is already there. For example, we look at the RNA scope or this paper, HCR3 or StarMap, they all use two different oligonucleotide sequences to achieve enhanced specificity. But when we look at the literature, we saw actually the design is very varied, right? Like which one is more optimized for specificity. And also they often couple this two probe idea with other complex amplification schemes. So we don't know which one of them actually is the best. And we also want to apply this coincidence detection idea to multiplex FISH. So we decided to go in and investigate ourselves and see which one of them worked the best. So the first thing we tried was that we reasoned that in order for this complementation hybridization idea to work, or this split probe idea to work, we need the probe to not fluoresce when it's by itself, and only fluoresce when two probes are together, right? So we first tested when this probe is by itself, whether it will give off fluorescence. And we actually tuned the length, the length of the dangling sequence or the wing sequence, or what we call here the split bridge sequence, to make it to vary its length. From here we varied from 7 nucleotides up to 12 nucleotides. And we see that when the nucleotide is longer, then it's already able to bind to the fluorescently labeled readout probes, right? But when the length is shorter, then it's not able to bind. So we found that we need actually the length to be 9 nucleotides or shorter for any individual split probe not to bind. And of course, to complete the optimization, we need to also see under what condition when two probes come together, then it can give off fluorescence. So we tested many ways, different ways to put the two probes together. We designed different ways to pair them. And actually, to our surprise, one of the way is very efficient at pairing the probes. And this structure here, this circular structure, we call it the SplitFISH, has actually as high efficiency as if we were to just bind a very long probe on a single probe. So we call this the conventional case. And we compared that they have almost the same efficiency. And this way of pairing the probes is actually the best among all the other configurations that we have tested. And finally, to finish off the optimization, we also try to tune the length because if the bridge sequence is too short, right, like a 7 or less, then even when we pair them, the probe will not be bright enough. So we found that 9 plus 9 nucleotide or 8 plus 8 nucleotide is the most optimal design. So with that design, we went in to do imaging to validate this probe design. we first tried to measure the specificity of the probes. We compare it with a conventional, but this time we use a lot of probes here as the conventional as a positive control, right? And by measuring the co-localization of the coincidence of two dyes, one is Psi3 where we mark the positive control, and one is Psi5 where we mark our split probe sequences and different design. So indeed, it agrees with our previous measurement that with this circular split probe, we can get the best specificity. And this is not surprising, right? Because they are the brightest. Also, because we not only want our probe to bind on target very well, we also needed to eliminate off-target signals. That's the whole point of us doing this in the first place, right? To make our images cleaner and able to scale to different tissue types without having to clear the sample. So as an example, we also tried to see if the SplitFISH can eliminate off-target signals. So we went to the literature and we found this very interesting example. So they also, this paper here, the authors were screening a lot of, were imaging a lot of non-coding RNAs, and they were very robust. So they were doing these two-color co-localization tests to check that their signals and their images are good, are accurate. So one very interesting example is that this non-coding RNA called XLOC010514, it failed the two-color co-localization test. And when they went in to investigate, they found that actually there is one oligo in the probe pool that is non-specific, right? So this particular oligo was staining a off-target in the nucleus. And when they removed this oligo, then the true localization of this non-coding RNA is revealed, right? And it's indeed in the cytoplasm. And now, when they stain the probe, half of them in Psi3 and half of them in Psi5, and then they co-localize well, indicating that this should be the true co-localization of true location of this non-coding RNA. So anyway, what we did was that we tried to see if with this SplitFISH design, we can eliminate these known off-target signals. So we first repeated the findings. So indeed, when we had this red oligo here, we saw the off-target signals in the nucleus. And when we remove these red oligos, the off-target binding in the nucleus has disappeared. But what is really encouraging is when we introduce the partner to all the probes, when we make them split probes, even with this off-target signal, the rogue probe is not, even with these off-target probes, this rogue probe is not able to give off-target signals because the misbinding of this sequence to the nucleus is by itself, and that off-target sequence will not have sufficient nucleotide, complementary nucleotide to allow the binding of the readout probes. So in that case, the split probe is indeed able to eliminate these off-target signals. So with that, I just want a quick summary that the circular configuration was able to improve specificity without sacrificing efficiency, and was able to reduce false signals associated with fish assays. And but of course, we want to see if can we adapt it to multiplex fish, right, which was our original goal to begin with. So we did more optimization to enable it to be to adapt it to oligo pool so that it can be in a big pool of 100,000 sequences. And then we design a specific sequential hybridization scheme. So where we first put in a library of encoding probe, And then we add this probe that we call the bridge probe that makes the coincidence detection. And then we flow in the dye-labeled readout probes to light up the RNA. So with this, actually, we were really surprised, that the background is really drastically reduced in multiplex fish. In fact, when we first saw it, we thought we must have mixed up the sample, that we're not actually staining a library. But the image indeed comes from this pool of oligo. And from here, you can see on the left here is when we stain with the conventional scheme where the probes are not paired. And then on the right here is when we stain with the SplitFISH scheme. So you see this reduction in background all around, and now the location of all the RNA is much clearer. So the sample almost looked like we have, cleared away all the backgrounds. And then we went on to optimize the buffer condition, and we showed that the SplitFISH is also able to show many rounds of sequential hybridization. In here, we show up to 20 rounds, and we still don't observe any degradation of the signal intensity or the RNA count. And what is really interesting is that because now we don't have to worry about how to clear the different sample types, we just take the same library and put it on different tissues. And we actually see all these very interesting and beautiful images of the different organization of the spatial transcriptome. For example, this one is the image of the brain, showing 4 out of the 317 genes that we see, and that we stain, and you can see this layer organization of the brain. Or this case here is a piece of the kidney tissue where we see this layer organization of the kidney as well. And this is the ovary tissue where we see different compartments of different cell types. And this is a piece of the liver where we see some cells outlining the blood vessels, and distinguishing between the portal and the central vein of the liver tissue. And we also did some technical validation where we compared the counts that we observed with the measurement from bulk RNA-seq. And, finally, we also see that compared in these uncleared tissues, compared to conventional RNA-seq, compared to conventional multiplex fish, the images, because it's clearer in SplitFISH, so we observe less false positive. And in the tissue, we also see, as I was mentioning, a different interesting organization of the tissue. For example, the cell layers that could, correspond to the flow of blood, for example, in the kidney, indicating maybe division of labor of the different cell types, and this idea of zonation in this tissue, or the compartmentalization that could, reflect the different developmental stages of the oocyte in the ovary. I just to conclude, I just want to mention, still a work in the progress. We have kind of try out different design, but this SplitFISH still has a limitation where the multiplexing capacity is reduced, right? If you compare it to the conventional. In the SplitFISH case, for the same region of the RNA, we not only have to put a pair of probe, but in one pair we can only put one barcode per probe pair, versus in the conventional case you can put up to 4 barcodes per pair. And I think this SplitFISH, more work needs to go into it to allows it to scale up to more genes. And we are also working on trying to, introduce signal amplification or tissue clearing to further improve the performance. With that, I just want to thank the people who really did the work to optimize the assay and the design. Specifically, Jolene, who did a lot of the experiments, and Nigel, who did a lot of the analysis. And with that, I would like to thank you all for coming to this seminar, and I would like to thank the funding agency, eStar from Singapore, and I'll be happy I'm going to pass the ball now to Alan, and after that, I think I'll be happy to answer your questions. Alan, please.
Thank you. I would like to complete this presentation by taking a very brief look at some of the kinds of detector solutions that are suitable for these kinds of experiments that have just been described. Different types of experiments that we do call for quite different requirements from a camera. So we might need a camera that emphasizes sensitivity or speed or field of view. So it depends what we're trying to achieve. And as of yet, no one camera is universally suitable for every experiment that we might want to try and do. And thus, there are many different camera models that we have to select from. But we can largely these days group these into two main technologies. Generally sCMOS cameras and also EMCCD cameras for certain applications. And these different technologies have certain strengths that make them more or less suitable for different types of imaging experiments that we might need to do. And over the years, these technologies have been continually refined, primarily trying to achieve improvements in sensitivity, but also with improvements in speed and trying to see larger fields of view as well. And in the research that has just been discussed and covered, they have used both EMCCD and back-illuminated sCMOS cameras, as we can see from a summary of two of the imaging setups. And in this case, the EMCCD camera was the Andor Exxon Ultra 888, and for the sCMOS camera, it was a Sona 4.2B-11 camera. So we'll briefly look at why these particular cameras would have been selected for the kinds of experiments that have been covered earlier. EMCCD cameras have long been known for the sensitivity that they can provide, and that remains very much true today. With the Exxon EMCCD cameras, they have 95% quantum efficiency, so 95% of photons are being collected and converted into electrons, but critically they also have EM gain, which we will mention. And these cameras also have deep cooling down as low as minus 100 degrees C, so that ensures the lowest noise floor, which is very important for EMCCD cameras. Less well mentioned with EMCCD cameras is that they have large pixels, and that allows them to collect more photons per pixel and allow for that better signal to noise ratio from the camera. And also the sensors in this case are in permanent vacuum sealed chambers to help allow them have that very low cooling and long-term stability. And as well, they also have the facility to run super resolution through any microscope using SurfStream Plus. There are two models of EMCCD cameras, or two sensors rather I should say. One of them is the Exxon 888, which has been covered here, and that has this larger array size for EMCCD cameras and the slightly smaller pixel. And there's also another model like 897, which is a smaller sensor format EMCCD. So since back-illuminated sCMOS cameras have been around for a couple of years now, people have made different comparisons of the sensitivity of the different cameras, and EMCCD cameras still remain the ultimate in sensitivity compared to any back-illuminated sCMOS camera, and that can easily be demonstrated. For example, if we consider looking at a very weak signal with 95% QE alone, we can end up getting no signal, but whenever we start to apply the electron multiplication, which EMCCD cameras, we can boost even single photons well beyond the noise floor of those cameras, so we're effectively defeating the read noise of the camera. So although they still have a read noise component, you have boosted up the signal so many times well beyond that noise floor, whereas at this level the back-illuminated sCMOS camera cannot work at all. So in terms of EMCCD cameras, we can see that they're clearly going to be suitable for the most challenging low-light applications. So we often generally say this as being for any of the single molecule detection studies, where these cameras would be the reference detector in those cases. And also virology for something that's a bit more current, but indeed anywhere where the amount of label that we're working with is low, that the EMCCD cameras can be used. In other cases, even for confocal, it can be very useful as well. And for longer exposures where we need to work with seconds to minutes to obtain a signal, the EMCCD cameras are the solutions to use, because there is up to 1,000-fold lower dark current than sCMOS cameras, so the sCMOS cameras would not be suitable once we're starting to move into seconds of exposures. And also for high-speed imaging, although sCMOS cameras, we talk about them as being very fast detectors, by running a camera very fast, the exposures are very short, and that means there's a very low number of photons. So what may be present on the spec sheet, sometimes in real life imaging, we may need to use an EMCCD camera to run a fast camera. However, we can be compromised in having a much smaller region of interest in the EMCCD cameras to achieve that. And in this work that has been presented, it was the Ixon Ultra 888 that was used, and that is the largest area EMCCD sensor and camera format that you can get, and it's actually the same sensor area as that common 4.2 megapixel sCMOS camera, so it's quite a large field of view from that camera. And with the deep cooling that camera has, it also allows you to have the lowest noise and deliver the best signal to noise ratio performance that is possible to get for those weakest signals. EMCCD cameras can be quite expensive, so there's also a lower cost version of those cameras called the Ixon Life series. So that's using the same sensors with similar specifications and really with a reduced feature set specifically for fluorescence microscopy. So there's some less options with that camera, but it allows you to access that sensitivity from the EMCCD cameras at a much lower price point. In terms of the back-illuminated sCMOS cameras, these latest sCMOS sensors bring back illumination, which, without going into the detail of that, which you can check from the Andor website, what this has the effect of is boosting the quantum efficiency of the sensor up to a similar level to the EMCCD cameras, up to around 95%. And that adds to all of the original strengths that sCMOS is known for, so the low noise, the high speeds, and the large sensor sizes. So it's no surprise that it's these types of sensors that are the most commonplace for general microscopy applications. And in terms of the Andor Sona cameras, the idea with these cameras was to extract the very best performance that's possible from those latest generation of sensors. And there's some of the ways that this is done is by using this permanent sealed vacuum technology, which is normally only found in those higher end Ixon EMCCD cameras. So that's the only back-illuminated sCMOS camera that has that type of technology built into it, and that helps allow for the deepest cooling. And with these cameras, they have a higher dark current, so it's very important to suppress that dark current from being an issue with those cameras, and also importantly reducing the amount of spurious hot pixels as well, and for the kinds of studies that have just been presented, we can see that it's very important to reduce any of these spurious events from affecting our data. Also importantly, these cameras have extended dynamic range, which allows them to handle the widest signal ranges. So that means that there's less need for adjusting camera settings, so that whenever you're imaging, you can tolerate a wide signal range and get optimum performance from the camera. And since many studies, like the one that's been shown, that quantitative accuracy is one of the fundamentals of any imaging cameras whenever we're trying to analyze that data. So beyond just the simple taking of the images, as we can see, it's very important to have a quantitative camera, and that has been one of the aims in these cameras as well, to make them as accurate as possible. So there's two sensor options available with that Sona camera series, and one of these uses the GS2020 BSI sensor, and the other uses the GS400 BSI sensor, and that's the one. The GS400 BSI sensor is the one that has been discussed in this work, and we can see just from comparing the specs in these, the Sona 4.2 B-6, which uses that 2020 BSI sensor, it has a smaller pixel size, but it's also low noise and high speed, so that's a very flexible camera, and it's the same sensor format as the previous generation of sCMOS cameras, so that's ideal for people looking to upgrade. However, the other camera that is covered has this larger pixel size, so although it's still 4.2 megapixels, the larger pixel size gives it this large sensor area, and that's what's particularly interesting about that sensor. The GS400 BSI sensor is all about this large sensor and large field of view, and we can really see that here whenever we can compare it in the Sona on the right with that large sensor compared to the other Sona, which has that same sensor format that we would see in the Xyla and the other older formats of sensor as well. So that large pixel along with the high QE is providing that high photon collection efficiency, for allowing for the high signal to noise ratio that we need in these kinds of experiments that have been discussed. And we can see this large field of view here, and not only is this much larger than the previous generation of sCMOS cameras and the Ixon, it is also much larger than competing cameras which have that same GS400 BSI sensor in it. So simply that allows you to capture more data. And also with this the Sona as well, it allows you to operate at longer exposures, because we don't always operate at very short exposures with sCMOS cameras for dealing with different labeling and experiments that we may do. So the deeper cooling allows for that low thermal noise, which is important for these sensors, and it cuts down those spurious and hot pixels that we really don't want to be getting in the way of any of the data analysis that we need to do. And one of the things it also has is this anti-glow, which tackles sensor glow, which is a problem with this sensor along with many others. So that is eliminated with this sensor, so you can get that full restricted field of view, which is the real importance of why you would want a GS400 BSI sensor. So the Sona 4.2 B-11 model is perfect for any experiments requiring high sensitivity over the widest possible field of view. And the other Sona with the smaller pixel, that's going to be more suitable for people working at lower magnifications where the smaller pixel is more suitable, and is a direct swap out for people who had the previous generation of sCMOS cameras. So thank you very much to Kok Hao and the rest of the team at the Genome Institute of Singapore for sharing with us today this very interesting research. And thanks to everybody for listening. And if you want to find out any more about the cameras available for your research, please check out the Andor website or contact myself. So thank you, everybody.
Thank you, Hao and Alan, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question that you would like to ask, please do so now. Just click on the Ask a Question tab located on the far left of your screen, and we'll answer as many of your questions as we have time for. So let's get started. Our first question is for Hao. So you mentioned using clearing methods can take days. About how long does this technique take to do?
Thank you for the question. Actually, it ties in nicely with the Andor talk as well. One of the factors that contributes to how long the assay takes is how large a field of view that we want to image. For example, in these tissue images where we look at like 1 to 2 millimeters square of areas, it could take up to half a day of imaging and washing and re-imaging, and to cycle through all this field of view. So yeah, having a large field of view cameras can shorten the assay time, but this can definitely can be done within a day or overnight at most. Depends on the number of genes and also the size of the tissue that we want to image. Yeah,
Great.
It's a very nice question. Mm-hmm.
Perfect. Thank you so much for that answer. So let's move on to our next question. So would adding tissue clearing to this technique provide any further benefit?
Yeah, I think it will, in the sense that it might further improve the signal to noise. It can, for example, reduce noise associated with the autofluorescence, like from protein or lipid, because these SplitFISH approaches focus on optimizing or rather increasing the specificity of the probe, right? The autofluorescence that comes along with the tissue will still show up in our images because we only so far focus on imaging in the red or the far red channel. The autofluorescence is relatively low, but if you use tissue clearing, for example, you can further reduce the autofluorescence of the tissue, especially in the blue channels, right, and allow us to do imaging there. So adding tissue clearing can definitely improve the quality of the assay. Yeah.
Wonderful. Thank you so much. And our next question, how much of the transcriptome can you cover with SplitFISH?
Okay. For this publication, right, we showed that we can image 317 genes. So that's maybe depends on how you estimate, like just a small 10-20% of the transcriptome. I think there are some technical hurdles, that we describe in the limitation slide that will allow us to scale to the whole transcriptome, and we are kind of working on that right now. And but in theory, the multiplex FISH approach, have already been shown by others that you can image the whole transcriptome. I thought that should be very exciting because it allows this unbiased approach for us to answer any kind of like phenotype genetics question that people have. Yeah. So right now we are at like about 300 genes, and we think maybe a few hundred more should be possible, but to get to the transcriptome scale, we need more tinkering.
Perfect. Thank you so much. So we do have time for a few more questions, but I do want to remind our audience that any questions that we are unable to answer today and those that come in during the on-demand period will be addressed by our speakers via the contact information that you provided at the time of registration. So moving on to our next question. Excellent method, thank you. Has there been any validation of this method on FFPE tissues?
Thank you. Thanks, Jamie, for the question, and it's a very good question. One of the motivation for developing this spatial transcriptomics is to look at the FFPE tissues, right? Because they are likely tumor tissues, and they could contain very important spatial information like immune infiltrates or tumor architecture. And we have in theory, the hybridization based assay is no different, whether you use it on mouse tissue or human tissue or FFPE tissue. I think the main challenge on the FFPE tissue is that many times the RNA in the tissue is degraded, so it will make it very difficult to stain them. Maybe the RNA is very broken down or already have very poor RNA quality. Then it will make the staining very challenging, and also the imaging as well. So I think the key to applying this on FFP tissue is firstly, have to deparaffin the tissue and maintaining the RNA quality, but also probably have to do a screen to verify that the RNA quality of the FFP tissue is still good before going ahead and profile its transcriptome. I think it's similar to other fish, fish approach on FFP tissue. But if you compare it to like RNA sequencing, maybe if you focus on like a 3' end or 5' end, then you might not need the full length transcript to be able to sequence it. So in that sense, sequencing the FFP tissue might give you a better output. Yeah. I hope that answers your question.
Wonderful, thank you. So our next question, I was wondering if you are planning to commercialize the SplitFISH technology or reagents.
Okay. Well, we don't have a way to like sell this reagents. But we can if you email us, we can help you to like design the probes and tell you where to buy them from. Yeah, and the reagents is actually very similar to standard RNA fish reagents, so you can probably make them yourselves. Yeah, so I hope that answers your question.
Wonderful, thank you. So it looks like we may have time for one more question. So are you able or have you tried to measure transcripts in a thick section, so 3D transcriptomics?
Okay. Yeah, that's a very nice question. I think this is certainly a goal. For now, because we are still we are using the standard wide field microscope to do this imaging. So very thick sections will be challenging to focus deeper. But if we can get rid of autofluorescence, for example, with some combining with clearing, or if we build or acquire more advanced imaging, like confocal or light sheet, then we can certainly try to image a thick section in 3D, and hopefully the dream is one day you can visualize the whole organ in situ, all the different cell types and what the different cells are doing, how are they connected to each other. I think that will be the dream. Yeah.
Great, thank you so much. So thank you again, Hao and Alan, for your presentations. Do either of you have any final comments for our audience?
No, just like to thank Hao again for presenting the work today. It's very interesting technique, and we can see that with the future development of it as well, it can have a very broad range of interest to people in the future.
Yeah, and thank you for the invitation, and thank you, Alexis, for hosting this talk, and thank you all for listening and coming to this webcast.
Thank you.
Before we go, I'd like to thank the audience for joining us today and for their interesting questions that we did not have time for today, and those submitted during the on-demand period will be addressed by our speakers via the contact information that you provided at the time of registration. We would like to once again thank our speakers for their time today and their important research. We would also like to thank LabRoots and our sponsor, Andor, for underwriting today's educational webcast. You can view the webinar on demand. LabRoots will alert you via email when it's available for replay. That's all for now. Until next time, bye-bye, everyone.
Bye-bye.